Number 2, our second speaker. It's really hard to go last. I'm really sorry, Brian. You gotta just. We have Raghav Yadavalli. He is a postdoctoral research scientist with the Bigelow Laboratory for Ocean Sciences. Raghav got his bachelor in technology and industrial biotechnology in 2008 from Jarwal. Did I get that right? I'm sorry. Nehru Technological Institute, India. Raghav moved to the US and obtained a PhD in regulatory biology with specialization in molecular and immunoparasitology at Cleveland State University. His thesis was focused on understanding how malaria parasite escapes the human immune system and causes devastating disease. Raghav's current research is focused on domesticating an oyster-infecting parasite, Parkinsus, thank you. I did read this, but I'm just not good at other words. Marinus, other languages, for vaccine production and immunization studies. And Raghav's current long-standing pop culture fandom is The Big Bang Theory. I think get a little closer.
Can everyone hear me now? Okay, I already lost. That's fine. So I work with oyster-infecting parasites, and I domesticate them for human immunization studies. Next, please. Oh. Can you use this? Okay. So how many of you like oysters here? So Maine is really big in culturing oysters and farming oysters, and it's one of the huge suppliers of oysters for food, right? People enjoy eating oysters. Oysters can be consumed by seasoning them, and there are a few people who love to eat oysters when they are raw. They just squeeze some lemon and then they eat it. Oysters do not only have nutritional benefits, but they are also useful. Recently, we did a huge research on how good the oysters are to be tested for biomedical research, and we showed that oysters have such cool properties in them where you can understand about cancer, you can understand about human immune system, a lot of things from them. And it made into social media quite a bit. And here are some of the examples. You can learn about the bone formation depending on understanding how the shells of the oysters are formed. And then you can learn about the immune system inside the humans by understanding like how oysters can survive even when they are exposed to so many viruses and bacteria when they are inside the water, right? They are the ones which will clear so much inside the water. So we can learn a bunch of things from oysters. But I work with the parasite which infects the oyster. It's called Parkinsus marinus, and it's highly prevalent on the Gulf of Maine coast. And we did PCR studies, and we did identify the infection of oysters being infected with this parasite. It's pretty predominant and it's pretty high. And these parasites can be cultured in the lab, can be grown in the lab without any oysters. I can grow these parasites inside a small flask by just providing food they need, right? My next logical question you guys might ask me is, do they infect humans, right? Oh, come on, I'm eating raw oysters. When I'm eating these, do these infect humans? We tried that experiment. We took immunized mice, and then we feed. You basically put it in water, put this parasite inside the water, taken from this culture, and you feed them. We do not see any infection. That's a good sign, right? It's wonderful. You do not see any infection. This does not infect humans. But most interesting thing that we identified is these parasites can actually trigger immune system inside the mice. So that means mice can identify this parasite as a pathogen and then can make immune response specific to that, right? So now we call this as an adjuvant, right? When you're putting a vaccine inside the humans, you always use a virus or a bacterial system to put the vaccine inside the body, right? So now I don't need a virus or a bacteria. I can use parasite by itself to deliver into the humans, right? So how do I do that? So I put the genes that I want or the vaccine candidates that I want to express inside the parasite, and then I electroporate that inside the parasites. If you can play this video for me, please. So this particular gene goes inside the parasite, we electroporate it, it tells me, oh yeah, that is inside the cell, and then I grow the cells. Now any cell which has this particular DNA inside it will show green color, which is like this. So this is the parasite which is showing the green color. That means this parasite is expressing the vaccine that I want it to express. So that's how I'm domesticating or cultivating this parasite to express the vaccine candidate inside them. So far, I cultured malarial vaccine candidate, toxoplasmosis, the cat disease, cryptosporidiosis, intestinal disease, and most interestingly, Ebola vaccine candidate inside the Perkinses. This is proof of concept. What am I going to do next? So now I know my parasite is expressing this protein. I'm going to manipulate the genome of the parasite using CRISPR-Cas9 technology. Most of you must have heard of it. You can ask me questions later. And then I'm going to decorate the parasite with the vaccine candidates on its surface so that your body can identify it well. And then grow those parasites and isolate the parasites which are expressing the green color that I have shown in the previous slide, and then culture them in the bioreactor with one of the students from Maine. Thank you.
So how many people are going to freak out when you tell them you're giving them a parasite?
I love this question a lot because I have been asked multiple times whenever I go to a conference, but here we are using this parasite because I do not need to irradiate the parasite. I do not need to expose them to any kind of isotopes to kill the parasite. I do not have to heat kill them, or I do not have to use any other virus thing which actually causes infection inside us. Rather, it is very safe, and I already have the data for that. And you know what's the best part is you don't have to inject this. You can just eat it. Like polio vaccines, if you have heard about it, like a couple of drops of polio vaccine, you get the vaccination against it, right? So that's how we are trying to develop this vaccine into. Instead of getting injected, it will taste good, like, you know, most of you do like oysters now, right? It's pretty safe, you can eat it. It's not going to cause any infection.
You can just use the microphone, that would be great, thank you. You can line up, you don't have to wait for me.
Yeah, just curious what sort of creature the parasite is. Is it a single-cell organism? Is it a multi-cell organism? What genus is it?
It's a single cellular organism. It belongs to alveolata. So it is a close relative to epicomplexans, which cause the other diseases that I showed, like malaria, toxoplasmosis, and cryptosporidiosis. So that's why I was able to successfully express that vaccine. Yeah. I have 3 minutes.
So what's the time frame getting this to actual use by humans?
That's a very good question. So right now I have the prototype, I have the proof of concept that I can express these vaccine candidates, but now once I synthesize these, I have to test them in mice, which are before even I can put them in the humans. So it has to go through those clinical trials, which might take 6 to 8 years, depending on how quick I can test it. And then by the time it gets to phase 3 or phase 4 clinical trials, that's when we are talking about trying this vaccine inside the humans. Yeah. So we are still due to test the vaccine in mice. Yeah.
Just around the corner, excellent.
Right.
All right, oh, go ahead, please.
Would this vaccine essentially, so I mean, it's a vaccine, so it's going to tell the body to replicate immune responses to whichever viruses, strands you've attached onto it? Is that essentially the goal?
Oh, yeah. So you see, each one of that is an antigen. So
Okay.
it's like a protein, recombinant protein vaccine. Unlike when you take H1N1, you take a viral vaccine by itself, which is the virus by itself, which is killed, heat killed, and then you put it inside you.
Right.
Here, we don't have to deal with any of those things. So no virus, nothing. It's just the protein with the parasite.
Oh,
So
okay.
you're exposing your immune system, the protein. You're not making your immune system to make the protein again.
I see.
So
Okay.
your immune system will generate antibodies specific to that protein once we put it.
Oh, thank you.
That's why it is much quicker and much safer. Yeah. Thank you.